Welcome to the deep dive. You've sent us quite the collection of research papers and lecture excerpts.
Yeah.
Everything from the intricate world of ion channels to
Yeah.
the history of the Nobel Prize and groundbreaking discoveries in physics, chemistry,
Environment.
biology, even economics.
Yeah.
We've got it
It's
all.
like a greatest hits of science, really
Yeah.
spanning decades of breakthroughs across all these different fields.
Yeah. Try to tie it all together in this
Yeah.
deep dive.
Yeah, I
So
like it to be fun.
We're on the hunt for those aha moments.
Yeah.
The surprising connections that make you see the world a little bit differently.
Exactly. We'll extract the key insights from each source.
Yeah.
But we'll also synthesize, find the common threads, help you grasp the bigger picture.
Yeah, I think one of those aha moments is going to be that that story about the scientist who gave himself an ulcer to prove.
Oh, yeah. Talk about dedication.
Talk about dedication.
Indeed.
That's pretty amazing.
And who knew you could mimic the behavior of crystals using just bricks and radio waves?
Bricks and radio waves.
We'll unpack all of that and more.
Okay, I'm intrigued.
But first,
Yeah.
to lay the groundwork,
Yeah.
let's start with something fundamental.
Okay.
Ion channels.
Ion channels, yeah.
This paper you sent on potassium ion channels.
Yeah. Yeah, this is fascinating stuff.
It's mind-blowing how these tiny little gatekeepers in our cells work.
Yeah, and I was reading about it, and it's not just a simple open and shut gate, right? It's much more complex than that.
Described as an ion water queue.
A carefully choreographed dance
Yeah.
that allows potassium ions to pass through while blocking others.
It's amazing how selective it is.
It is, yeah, very selective.
So how did scientists even figure that out?
Right.
I mean, these channels are way too small to even see.
Well, this is where another Nobel Prize-winning discovery comes.
Right.
X-ray crystallography.
Oh, right.
This allowed researchers to actually map the channel's structure at the atomic level.
So seeing this precise arrangement of atoms helps
Yeah.
explain how the channel optimizes potassium ion flow.
It's pretty incredible how
Mm.
they were able to figure that out.
Mm.
What I found really cool was the paper's mention of electrostatic repulsion.
Oh,
Like the
yeah.
potassium ions are pushing each other along.
Right. It seems counterintuitive,
Yeah.
but that repulsion is actually crucial for achieving those super fast conduction rates.
Yeah.
It all comes down to the interplay of multiple factors, the
Yeah.
structure, electrostatic forces, even the presence of water molecules.
Yeah.
Very intricate.
This potassium channel study is fascinating on its own.
Yeah.
But it makes me wonder, are there similar mechanisms at play in other types of ion channels?
That's a great question, and it's a question that researchers are actively exploring.
Oh, cool.
There's a whole family of ion channels out there,
Right,
each
right.
with its own specific role
Yeah.
and selectivity for different ions. So what we learn about one can often shed light on the workings of others.
It's like a blueprint that they can then use to understand the others.
Yeah, it's a good analogy.
It's a reminder that even the seemingly simple processes in our bodies
Right.
are incredibly complex,
Incredibly complex and finely tuned.
finely tuned.
Yeah.
And speaking of breakthroughs that illuminate complexity,
Yes.
one of these Nobel lectures describes a game-changing technique called mass spectrometry.
Oh, yes, mass spectrometry.
Mass spectrometry. I remember reading about its use
Mm-hmm.
in identifying and analyzing proteins,
Right.
even those involved in forming ion channels.
Exactly.
So this was
Koichi Tanaka
Koichi Tanaka.
was the Nobel laureate behind this breakthrough.
Yeah.
And he pioneered a way to use lasers to ionize large molecules.
So they're using lasers on
Yeah.
molecules now.
It's like shining a light on the molecular world,
Wow.
allowing us to study these large, complex molecules like never before.
So this laser technique wasn't just an advancement in physics.
No, not at all.
It had a ripple effect across multiple disciplines,
Absolutely.
including our understanding of those crucial ion channels
Right?
we were just talking about.
Bringing it all back around.
Yeah.
Full circle.
It underscores a theme we'll see again and again.
Mm-hmm.
Advances in one area can unlock entirely new possibilities in others.
Absolutely. That's the beauty of science.
Yeah.
It's all interconnected.
This is already making me appreciate the interconnectedness of scientific discovery.
That's great to hear.
And that interconnectedness goes beyond just different fields of science.
Right.
Let's take, for example, atomic clocks.
Oh, yes. Atomic clock.
Atomic clocks, the most accurate timekeepers we have.
Mm-hmm.
But I've always been a bit fuzzy on how they actually work.
Well, imagine harnessing the incredibly specific frequencies at which atoms absorb and emit light.
I see.
That's the heart of an atomic clock.
Okay.
So you're using the properties of atoms to keep time.
Okay, I'm following so far.
Okay.
Where do lasers come into play?
Ah, that's the ingenious part.
Okay.
You see, atoms are constantly jiggling around due to their thermal energy.
Uh-huh.
This introduces uncertainties into those frequency measurements,
Right.
which makes the clock less accurate.
I see. Yeah.
So how do you get around that?
Right. How do they solve that?
They use lasers to cool those atoms down.
Wait,
It might seem counterintuitive,
I was just going to say.
but by carefully tuning lasers to specific frequencies, scientists can essentially chill the atom.
So lasers, which we usually think of as incredibly hot, can also be used to cool things down.
That's right.
That is mind-blowing.
It's a really cool application of physics.
Yeah.
And it really underscores the importance of challenging our assumptions about what's possible.
Yeah, absolutely.
Because our intuition can often lead us astray.
I never realized something as seemingly abstract as laser cooling could have such a direct impact on something as everyday as timekeeping.
It's a hidden technology that underpins everything from GPS navigation to fundamental scientific research. It's
amazing.
Mind-blowing to think how a discovery in atomic physics can have such far-reaching consequences.
It really can.
Okay, we've gone from the microscopic world of cells to the even tinier world of atoms.
Yeah, what a journey.
I'm excited to see where this deep dive takes us next. But before we jump into a new topic, how about we take a moment to reflect on what we've learned so far?
Sounds good.
What stands out to you from this first part of our journey?
Well, for me, it's that reminder that even the most basic processes in our bodies, like the movement of ions across cell membranes, are just incredibly complex and finely tuned.
They are, and they have to be right.
They do.
Otherwise, things would go haywire.
Yeah.
And the fact that we can understand these processes at the atomic level.
I know, right?
Thanks to techniques like X-ray crystallography.
It's just mind-blowing.
Mind-blowing, yeah.
It really is.
Yeah, and I think it's also fascinating how advancements in one field, like Tanaka's work on mass spectrometry, can have these ripple effects across multiple disciplines, pushing forward our understanding in seemingly unrelated areas.
Exactly. And I think the story of the atomic clocks and laser cooling really illustrates that as well, right?
Absolutely.
Who knew that something so seemingly abstract could have such profound impact on our everyday lives?
Right. You don't think about it, but it's there.
It's just there in the background. It really speaks to the power of basic research.
It does.
And the importance of exploring the unknown, even if the practical applications aren't immediately apparent.
Exactly. Curiosity-driven research is so important for pushing the boundaries of knowledge.
Absolutely.
Yeah.
Okay, I'm ready for the next adventure.
All right.
Where to next on our scientific journey?
Well, how about we shift gears from the microscopic to the macroscopic?
Okay.
From the world of atoms and cells to the realm of economics.
Economics, okay.
One of the Nobel lectures we have is from Kenneth Arrow, who made groundbreaking contributions to general economic equilibrium theory.
Okay. Economics. Now, that's a bit of a jump.
It is a bit of a jump.
From ion channels and laser-cooled atoms.
Yeah, we're going from the very small to the very large.
Yeah.
But that's the beauty of this deep dive, right?
Right. We get to explore
all these different areas.
Yeah. What exactly is general economic equilibrium theory?
Right.
And why is it important?
Okay, so it's a complex field.
Yeah.
But in essence, it tries to explain how the actions of individuals, companies, and governments all interact to determine things like the prices of goods and services and how resources are allocated.
So it's like trying to understand how the whole economic machine works.
Yeah, that's a good way to put it.
That sounds incredibly ambitious.
It is very ambitious.
Yeah.
And Arrow's work was particularly important in figuring out under what conditions a competitive market could actually lead to an efficient allocation of resources.
What does that mean, efficient allocation of resources?
No, this is a situation where nobody can be made better off without making someone else worse off.
So that sounds like a pretty ideal scenario.
It does, yeah.
But does it really hold up in the real world?
Right.
Where markets aren't always perfectly competitive?
That's a great question.
Yeah.
It's one that economists continue to debate to this day.
Oh, okay.
Arrow himself acknowledged that real-world markets are often messy.
Yeah. Yeah, definitely.
With things like monopolies, hidden costs, unequal access to information.
Yeah, lots of things that get in the way.
Lots of complexities.
So his work was more about establishing a theoretical framework than perfectly describing reality.
I think that's a fair assessment, yeah.
Okay, but that doesn't diminish its significance, right?
No, not at all.
I mean, by laying out a rigorous theoretical foundation, Arrow's work gave us a benchmark.
Exactly.
To compare real-world markets
against. A standard to measure against.
Right. It helps us identify where inefficiencies exist.
Right, where things aren't working as well as they could.
Right.
Yeah.
And potentially how to address them.
Exactly.
I can see how that would be valuable for policymakers trying to create a more equitable and functional economy.
Absolutely.
So
his work has had a big impact on policy.
It's applicable to the real world.
Yeah, it's not just theoretical. Yeah. It has real-world implications.
And Arrow's contributions went beyond just market economics.
They did.
He also delved into social choice theory, exploring how groups make decisions.
Right, whether it's voting systems or how companies are governed.
So this Nobel lecture isn't just about economics in the traditional sense.
No.
It also offers insights into the dynamics of decision-making.
Absolutely.
In any group setting.
Yeah, in any group setting, yeah.
It's fascinating to see how these seemingly abstract economic theories connect to our everyday lives.
It is, yeah. It brings economics down to earth.
This deep dive is truly showing me how interconnected everything is.
That's what we're hoping to achieve.
And speaking of interconnectedness,
yeah.
Remember those ion channels we discussed earlier?
Oh, yes. The gatekeepers.
Let's talk about another powerful tool that's revolutionized our understanding of them. NMR spectroscopy.
NMR spectroscopy, yes.
Wait, NMR, isn't that the same technology behind MRI scans?
You got it. NMR stands for nuclear magnetic resonance.
Right.
And it's a technique that can be applied in both medicine and fundamental research.
Right.
The Nobel lecture we have is from Kurt Wuthrich,
Wuthrich,
who revolutionized the use of NMR to determine the structures of proteins, including those that make up ion channels.
So, before Wuthrich's work, how did scientists figure out the 3D structure of proteins?
Well, it was a much more laborious process.
I can imagine.
It often involved crystallizing the protein,
Yeah.
which could be incredibly time-consuming and didn't all work.
Right, and so they had to have huge amounts of the protein.
Exactly.
But
Wuthrich changed the game.
How did he do that?
He figured out how to study proteins in their natural dissolved state.
So you didn't need to crystallize them anymore?
No, which was a huge advantage.
Yeah.
But how does NMR actually work?
Yeah, I mean, it sounds pretty high-tech.
It is high-tech.
Yeah.
It leverages the magnetic properties of atomic nuclei within the protein. So by analyzing how these nuclei respond to radio waves in a magnetic field, Wuthrich could deduce the protein's 3D structure in solution.
So it's like using radio waves to create a 3D map of the protein.
That's a good analogy.
That's incredible. And this opened up a whole new avenue.
It did, yeah.
Of understanding how proteins function in their natural environment, right?
Precisely. It revolutionized structural biology.
Wow.
Allowing us to study proteins involved in everything from cell signaling.
So it has a wide range of
processes,
applications.
A wide range of applications, yes.
And all this stemmed from a technology initially developed for physics research.
That's right. Another example of that interconnectedness we were talking about earlier.
This deep dive is making me realize how much of science is built upon adapting and refining existing techniques.
It's a constant process of innovation.
Yeah.
Building on what came before.
And sometimes those adaptations lead to breakthroughs in entirely unexpected areas.
Absolutely.
That's exciting.
It is, yeah.
Okay, so we've explored the intricate world of ion channels. A technique for analyzing complex molecules.
Yeah.
And even delved into economics and decision-making.
Yeah, quite a range of topics.
Yeah. I can't wait to see what other scientific gems are waiting to be discovered.
There are plenty more to come, I assure you.
Okay.
It's a powerful tool for building molecular rings.
Okay.
Which are like the scaffolding of many organic molecules. The Diels-Alder reaction is a chemist's go-to for creating these rings
Yeah.
with elegance and precision.
Now I remember those ring structures were everywhere in organic chemistry.
Mm-hmm.
And we have a Nobel lecture on this reaction.
We do.
Oh.
It's from Kurt Alder himself, who shared the Nobel Prize in Chemistry in 1950 with Otto Diels for their discovery and development of this remarkable reaction.
So this lecture takes us back to a time when chemists were developing those fundamental tools for building the complex molecules we rely on today.
Exactly.
I'm curious.
Yeah.
What are some real-world examples of where the Diels-Alder reaction has been used to create important molecules?
Well, one classic example is the synthesis of insecticides, like aldrin and dieldrin, which were widely used in the mid-20th century. The Diels-Alder reaction was key to constructing the complex ring systems in these molecules.
So this isn't just some abstract chemical reaction.
No, not at all.
It's had a real-world impact.
Absolutely.
Helping to control pests and protect crops.
It has, yeah.
And its applications go far beyond insecticides.
Oh, absolutely.
Right.
The Diels-Alder reaction has been used to synthesize countless pharmaceuticals, natural products, and other crucial molecules.
It really highlights how a fundamental understanding of chemical reactions can lead to practical solutions for real-world problems.
It's a great example of that, yeah.
It's like having a molecular toolkit.
I like that analogy.
For building almost anything.
Like a molecular toolkit.
Yeah.
And speaking of toolkits, how about we explore the discovery of a remarkable enzyme called ribonuclease P.
Ribonuclease P.
It plays a crucial role in processing RNA molecules, which we know are essential for carrying genetic information and doing all sorts of jobs in the cell.
Ribonuclease P.
Yeah.
Didn't we already talk about RNA when we discussed Andrew Fire's work on RNA interference?
We did. And this discovery adds another layer to our understanding of RNA. You see, RNA molecules often need to be processed and modified before they can do their jobs properly.
Right.
And ribonuclease P is a key player in this processing, specifically in the maturation of transfer RNA.
Transfer RNA.
Or tRNA molecules.
Okay, tRNA.
Yeah.
Can you remind me what tRNA molecules do again?
Oh, sure.
I remember they're involved in protein synthesis, but the details are a bit fuzzy.
Well, think of tRNA molecules as the delivery trucks of the cell.
The delivery trucks.
Yeah, they carry specific amino acids to the ribosomes where proteins are assembled.
Ah, right. So ribonuclease P helps to get those tRNA molecules ready for their crucial role in protein synthesis.
Exactly. It's preparing them for their job.
And we have a Nobel lecture on this enzyme.
We do. It's from Sidney Altman.
Sidney Altman.
Who, along with Thomas Sick, received the Nobel Prize in Chemistry in 1989 for their discovery of the catalytic properties of RNA.
Hold on. I thought enzymes were proteins.
Right. That was the thinking at the time.
Yeah.
What do you mean by the catalytic properties of RNA? That's what makes this discovery so revolutionary. Up until Altman and Sick's work, it was a central dogma of biology that only proteins could act as enzymes.
Yeah.
But they turned that dogma on its head.
Okay, so you're telling me that ribonuclease P is made of RNA, not protein.
That's right.
And it acts as an enzyme.
Exactly.
Whoa.
It's called a ribosome. Yeah, a type of RNA molecule that can catalyze biochemical reactions.
So it's like a protein enzyme, but made of RNA.
Exactly. A very special type of RNA.
Wow, that's incredible.
It is, yeah.
It challenges everything I thought I knew about enzymes.
I know. It's a really mind-bending discovery.
Yeah.
So RNA is not just a passive carrier of genetic information.
Right.
It can also play a very active role in cellular processes.
Right. It's not just a blueprint. It's actually doing things.
It's a worker, too.
Yeah. This is mind-blowing.
Oh, it is, yeah.
And the discovery of ribozymes, like ribonuclease P, has opened up new avenues of research.
It has, yeah.
Right.
Not just in understanding how RNA works, but also in potentially harnessing these ribozymes for therapeutic purposes.
So there could be medical applications of this discovery.
That's the hope. Yeah.
It makes you wonder what other surprises RNA has in store for us.
It does. There's still so much we don't know about RNA.
Yeah.
It's a fascinating molecule.
And speaking of surprises,
how
about we delve into the groundbreaking work of Stanford Moore and William Stein on the structure and function of ribonuclease.
Ribonuclease,
a protein enzyme that degrades RNA molecules.
All right, so now we're looking at the other side of the coin.
Ribonuclease.
Yeah.
Wait a minute. Isn't that the opposite of what ribonuclease P does?
That's a great observation.
Okay.
While ribonuclease P is involved in processing and maturing RNA molecules, ribonuclease, often called RNase, is responsible for breaking them down.
So we're exploring two sides of the same coin.
Exactly.
The synthesis and degradation of RNA molecules.
It's all about balance, right?
Right.
You need both processes to maintain proper cellular function.
And Morenstein's work on RNase was groundbreaking for several reasons.
It was.
Right.
They were among the first to determine the complete amino acid sequence of a protein.
The complete amino acid sequence.
Yeah, essentially providing a blueprint of its molecular structure.
So their work was crucial in establishing the link between a protein's amino acid sequence, its building blocks, and its three-dimensional structure.
And ultimately its function. Yeah, it's all connected.
And their meticulous research on RNase's mechanism of action revealed how this enzyme specifically recognizes and cuts RNA molecules.
Right, they really delved into the details of how it works,
providing valuable insights into the principles of how enzymes work.
It was a major contribution to our understanding of enzymology.
This deep dive is reminding me that to truly understand biological processes, we need to look at both sides of the equation.
Exactly.
The building up and the breaking down of molecules.
It's all about maintaining that delicate balance.
It's all the delicate balance.
Yep.
Yeah.
And speaking of balance, how about we shift our attention to the work of Earl Sutherland, who discovered a key molecule involved in cellular signaling.
Okay.
Cyclic AMP.
Cyclic AMP.
Or AMP, as it's often called.
Cyclic AMP. That rings a bell in my biology class, but I'm a bit rusty on the details.
Okay, well, think of it as a cellular messenger.
Okay.
Relaying signals from outside the cell to inside the cell.
A cellular messenger.
It helps cells respond to hormones and other external cues.
I see.
Ensuring they're doing the right thing at the right time.
So it's like a molecular communication system within the cell.
That's a good way to put it, yeah.
What makes cyclic AMP or KMP
so special?
Well, Sutherland's discovery of KMP was groundbreaking.
Okay.
Because it revealed a fundamental mechanism by which cells communicate and respond to their environment.
So it's like a universal language that cells use to talk to each other.
Yeah, a way for them to send and receive messages.
Wow. That's a pretty big deal. And we have a Nobel lecture on this discovery.
We do. It's from Earl Sutherland himself.
Okay.
Who won the Nobel Prize in Physiology or Medicine in 1971.
Right.
For his discoveries concerning the mechanisms of how hormones work.
Right.
Specifically, his work on KMP.
So this lecture takes us back to a time when scientists were just starting to unravel those complex communication networks.
It does, yeah.
Within cells.
It was a time of great discovery in cell biology.
It must have been like deciphering a secret code.
It was very much like that, trying to figure out how cells talk to each other.
Yeah. What were some of Sutherland's key findings about how this cellular messaging system works? Well, one of the crucial findings was that KMP is generated from ATP, the cell's primary energy currency, by an enzyme called adenylyl cyclase.
Adenylyl cyclase, okay.
And this enzyme gets activated by various hormones and neurotransmitters, leading to a surge in KMP levels within the cell.
So it's like a cascade of events. A signal from outside the cell triggers a series of molecular changes inside the cell.
Exactly.
Ultimately leading to a specific response.
That's how signaling pathways work.
What kind of responses are we talking about?
Well, everything from changes in metabolism to alterations in gene expression.
Right.
Sutherland's work revealed that KNAMP activates a key enzyme called protein kinase.
Protein kinase A.
Which in turn modifies and regulates a whole slew of target proteins.
Oh, wow.
Leading to a wide range of cellular responses.
This deep dive is highlighting the incredible complexity of cellular signaling.
It is, yeah.
It's amazing how our cells are constantly receiving and interpreting signals from their environment.
Never busy.
And orchestrating a symphony of responses.
It's like a beautifully orchestrated dance happening inside every cell.
To maintain balance and function.
Exactly.
And speaking of revolutionary technologies, let's shift gears back to the realm of electronics and delve into William Shockley's Nobel lecture on transistor technology.
Transistor technology, yes.
Transistor technology.
Yeah.
Didn't we already discuss transistors when we talked about John Bardine's Nobel lecture?
Here we did.
Okay, so...
But Shockley's lecture offers a different perspective.
A different perspective.
While we explored the invention of the transistor and the scientific principles behind it, Shockley focuses more on the evolution and impact of transistor technology.
Okay.
Particularly its contributions to physics.
Ah, okay. So we're going beyond just the invention itself and looking at how transistor technology has shaped our understanding of the physical world.
Exactly.
That's an interesting angle.
It is. Shockley's lecture highlights how the development of transistors led to new insights into the behavior of electrons in solids, particularly in semiconductors.
It's fascinating how a technology that revolutionized electronics also ended up deepening our understanding of the fundamental properties of matter.
It's a great example of how technology can drive scientific discovery and vice versa.
And Shockley's work was instrumental in establishing the field of solid-state physics.
Absolutely.
Which underpins so much of our modern technology.
It does, yeah. It's the foundation for so many things.
His lecture also mentions the concept of deathnium.
Oh, yes, deathnium.
Right. It sounds a bit ominous.
It does, doesn't it?
Yeah.
But it's actually a term he coined to describe a type of imperfection in a crystal lattice that can trap electrons, affecting the conductivity of the material.
So it's like a tiny defect in the otherwise perfect arrangement of atoms in a crystal.
Yeah, that's a good way to think about it.
That can disrupt the flow of electrons.
Exactly.
Okay, so it's not...
It's a bit of a whimsical term.
Yeah, it's not as scary...
It's for a serious concept.
Yes, it sounds.
Yeah.
But it's important.
But it is an important concept because these imperfections can have a big impact on how semiconductors behave.
Right.
Shockley's insights into the role of imperfections in semiconductors were crucial for improving transistor performance and developing new semiconductor materials.
This is a great example of how understanding the nuances of a material, even its imperfections, can be crucial for harnessing its full potential. It's about understanding how things work at the most fundamental level.
Exactly. It's all about the details.
And speaking of fundamental discoveries, how about we venture into the world of nuclear physics?
All right, let's do it.
And explore Maria Goeppert-Mayer's Nobel lecture on the nuclear shell model.
The nuclear shell model.
The nuclear shell model.
Right.
I'm not familiar with that one.
Okay, well...
Can you give me a crash course?
Imagine the nucleus of an atom.
Okay.
That tiny, dense core made up of protons and neutrons.
Right, right.
The nuclear shell model is a way of understanding how those protons and neutrons are arranged within the nucleus.
I see.
It proposes that they occupy distinct energy levels, or shells.
Oh.
Much like electrons occupy shells in an atom.
So we're talking about the internal structure of atomic nuclei.
Exactly.
How those fundamental building blocks are organized.
Yes. It's a way of understanding the architecture of the nucleus.
Okay.
And the nuclear shell model was a breakthrough in understanding the behavior of nuclei.
Right.
It successfully explained many of the observed properties of nuclei.
Such as
their stability, their spin,
right,
and their magnetic moments.
So things that had puzzled scientists?
Things that had puzzled scientists for a long time.
For a long time.
Yeah, it was a major step forward.
And we have a Nobel lecture on this model.
We do.
Okay.
It's from Maria Goeppert-Mayer herself.
Maria Goeppert-Mayer.
Who shared the Nobel Prize in Physics in 1963 with J. Hans D. Jensen for their independent development of the nuclear shell model.
This deep dive is not only shedding light on incredible scientific discoveries, but also celebrating some of the brilliant minds behind them.
It is, yeah. It's a celebration of human ingenuity and curiosity.
Yeah, it's really inspiring.
It is.
So back to the nuclear shell model.
Okay.
What were some of the key features of this model that Goeppert-Mayer emphasized in her lecture?
Well, one of the most important features was the introduction of a strong spin-orbit interaction.
Spin-orbit interaction.
Yeah, this means that the energy levels of protons and neutrons are affected by their intrinsic angular momentum.
Right. So it's not just about the spatial arrangement of those protons and neutrons.
No, it's not just about where they are.
Within the nucleus.
Their internal spin also plays a role in determining their energy levels.
It's incredible how even at the subatomic level, there are these intricate layers of complexity and interplay between different forces.
Yeah, it's a very intricate dance happening at the heart of matter.
And the nuclear shell model, well, A simplification of reality.
Right. It's a model.
Provides a powerful framework.
But it's a very useful model for understanding the behavior of atomic nuclei.
This deep dive is really highlighting the power of theoretical models in science.
Absolutely. Models are essential tools for understanding complex systems.
They provide a lens through which we can view and make sense of complex phenomena.
Yeah. They help us to simplify.
Even if they can't capture every nuance.
And make sense of the complexity.
Right, right.
Yeah.
Okay, where does this scientific adventure take us next?
How about we explore the work of Wolfgang Paul?
Okay.
Who developed a technique for trapping charged and neutral particles.
Trapping particles.
Using electromagnetic fields.
With electromagnetic fields. That sounds pretty futuristic.
It does sound futuristic, doesn't it?
Yeah.
But it's a real technique that has revolutionized our ability to study and manipulate particles at the atomic level.
Can you paint a picture of what that looks like?
Sure.
And why it's so important?
So imagine creating a tiny invisible cage made of electric and magnetic fields to trap individual particles.
Wow.
Like ions.
So they're using
these devices are called ion traps.
Ion traps.
Yeah, and they have revolutionized our ability to study and manipulate particles.
Sounds like something straight out of science fiction.
It does. It's pretty amazing technology.
Yeah.
But it's real.
Yeah.
And it's led to some incredible advancements in our understanding of the universe.
And we have a Nobel lecture from Wolfgang Paul himself.
We do. Who shared the Nobel Prize in Physics in 1989.
Wow.
With Norman Ramsey and Hans Dehmelt. For their work on developing techniques for trapping and studying atoms and ions.
So this lecture delves into the world of precision measurement?
It does, yeah.
And control at the atomic scale?
It's all about manipulating and measuring things at the tiniest level.
I'm curious, what are some specific examples of how ion traps have been used to push the boundaries of scientific knowledge?
Well, one of the most remarkable applications is in atomic clocks.
Atomic clocks?
The most accurate timekeepers ever invented.
Right. I remember you were talking about those earlier.
Ion traps are used to confine and cool ions to extremely low temperatures, slowing their motion, and allowing for incredibly precise measurements of their atomic transitions.
So they're basically freezing them in place.
Essentially, yes.
So they can measure them more accurately.
Yeah. It's all about reducing those uncertainties we talked about earlier.
Wow. So Paul's work on ion traps has indirectly contributed.
It has, yeah.
To the accuracy of our GPS systems and other technologies that rely on precise timekeeping.
Absolutely.
Pretty mind-blowing.
It's pretty amazing.
And the applications of ion traps extend far beyond atomic clocks.
Oh, absolutely.
Right.
They're used in mass spectrometry to identify and analyze molecules.
Right.
In quantum computing to manipulate qubits.
Qubits?
Yeah. And even in biological research to study the structure and function of proteins.
So they have a wide range of applications.
Yes, they do, yeah.
It seems like ion traps are an incredibly versatile tool.
They are.
With applications across a wide range of scientific disciplines.
And it highlights another recurring theme in this deep dive. The interconnectedness of scientific knowledge.
Right.
And how advancements in one field can have a ripple effect across many others.
Yeah, absolutely. Okay, I'm ready for another adventure.
All right, let's go.
Where will this exploration take us next?
Well, how about we shift our focus from the microscopic world of atoms and ions to the fascinating realm of vision and neurobiology.
Okay, vision. We have a Nobel lecture from H. Keffer Hartline.
H. Keffer Hartline.
On visual receptors and retinal interaction.
Retinal interaction.
Visual receptors and retinal interaction.
So we're diving into the workings of the eye and the brain.
I've always been fascinated by how our senses work.
Yeah, it's amazing.
Especially vision.
It's how we perceive the world around us.
Yes.
It's a very complex process.
It's a very complex process.
Hartline's work focused on understanding how our eyes detect light signals and how those signals are processed by the nervous system to create our sense of vision.
And we have a Nobel lecture on this very topic.
We do. It's from H. Keffer Hartline himself, who shared the Nobel Prize in Physiology or Medicine in 1967 with Ragnar Granit and George Walt for their discoveries concerning the primary physiological and chemical visual processes in the eye.
It's incredible to think
It is.
we can understand something as complex as vision at such a fundamental level. What was Hartline's approach to studying vision?
Well, his research focused on the horseshoe crab.
The horseshoe crab.
Which has a relatively simple visual system compared to ours.
Oh, I see.
This made it an ideal model.
They're using a simpler system.
For studying the basic principles of vision.
To understand the more complex one.
Exactly.
So, by studying a simpler system, he was able to gain insights into the fundamental mechanisms of vision.
That's the idea.
That apply across species.
That's right. It's a common approach in science.
Yeah, it makes sense.
Studying a simpler system to gain insights into more complex ones.
What were some of the key insights
that
Hartline's research on the horseshoe crab revealed?
Well, one of the key findings was that individual photoreceptor cells in the eye, the cells that actually detect light, respond to light with a burst of electrical signals.
So each photoreceptor cell acts like a tiny light sensor.
Yeah, that's a good analogy.
Sending signals to the brain when it detects light.
Exactly. It's converting light into electrical signals.
But it's not just about individual cells working in isolation.
No, it's not just about individual cells.
Right.
Hartline's research also revealed that these photoreceptor cells are interconnected in a way that allows them to influence each other's responses.
Ah, so the signals from individual photoreceptors aren't just sent directly to the brain.
No, there's more to it than that.
There's some processing and integration happening within the retina itself.
That light-sensitive layer at the back of the eye.
Right, right.
Hartline discovered a phenomenon called lateral inhibition.
Lateral inhibition.
Where the activity of one photoreceptor cell can actually inhibit the activity of its neighbors.
So one cell can tell its neighbor to quiet down.
Essentially, yes.
That's so interesting.
It is, yeah.
Why would they do that?
Well, it might seem counterintuitive.
Yeah.
But it actually enhances contrast and sharpens the visual image.
Oh, I see.
So it's a way of refining the visual information before it even gets to the brain.
So our eyes are doing more than just passively detecting light.
They are, yeah.
There's a whole lot of signal processing going on.
It's a very active process in the retina to refine and enhance the visual information before it even gets to the brain.
It's amazing how much complexity is packed into something as seemingly simple as seeing.
It really is. Vision is a marvel of biology and physics.
And speaking of amazing phenomena, how about we explore Ivar Jeever's Nobel lecture on the phenomenon of tunneling in superconductors.
Tunneling in superconductors.
Tunneling in superconductors.
All right.
Now that sounds like we're venturing into the realm of quantum mechanics again.
It does, yeah.
But first, can you remind me what superconductors are?
Okay.
I know they have something to do with electricity, but the details are a bit fuzzy.
Well, think of superconductors as materials that can conduct electricity with zero resistance.
With zero resistance.
So it's like a superhighway for electrons.
Wow.
Where they can flow without any loss of energy.
Wow, that's incredible.
It is, yeah. It's a very special property.
But what does tunneling have to do with superconductors?
So tunneling is a quantum mechanical phenomenon where particles like electrons can pass through barriers that would be impossible for them to cross according to classical physics.
Right. It's like they can magically teleport through walls.
It's a very strange phenomenon.
It's very strange.
But it's a real phenomenon.
Quantum mechanics never ceases to amaze me.
It's full of surprises.
So how does tunneling manifest in superconductors?
Well, Jeever's work demonstrated that electrons can tunnel through thin insulating layers, separating two superconductors. This seemingly impossible feat provided crucial evidence for the theory of superconductivity.
So it helped to confirm the theory.
It did, yeah. It was a key piece of the puzzle.
And we have a Nobel lecture on this very discovery.
We do. It's from Ivar Jeever himself.
Ivar Jeever.
Who shared the Nobel Prize in Physics in 1973. With Brian Josephson and Leo Asaki. For their experimental discoveries regarding tunneling phenomena in semiconductors and superconductors.
So this lecture takes us back to a time when physicists were grappling with the mind-boggling world of quantum mechanics.
It does, yeah.
And its implications for material science.
It was a time of great excitement and discovery in the field of condensed matter physics.
What were some of the key implications of Jeever's discovery? Did it have any practical applications?
Well, his work not only confirmed the theory of superconductivity, but it also paved the way for the development of incredibly sensitive devices, such as superconducting quantum interference devices.
Superconducting quantum interference devices.
Or SQUIDs.
SQUIDs, okay.
These devices are used in a wide range of applications.
Okay, so...
from medical imaging to geological exploration.
So this fundamental research into a rather esoteric phenomena ended up having a tangible impact on technology.
It did, yeah.
And our ability to study the world around us.
Sometimes the most unexpected discoveries come from exploring.
That's pretty amazing.
Those seemingly bizarre corners of the universe.
It is.
It underscores the importance of curiosity-driven research.
Yeah.
Yeah.
Okay, I'm ready for another scientific adventure. What's next on our agenda?
How about we shift gears from the world of physics to the realm of medicine?
Okay, medicine.
And explore Barry Marshall's Nobel lecture on the discovery of Helicobacter pylori.
Helicobacter pylori.
And its role in gastric ulcers.
Ah, Helicobacter pylori.
Yeah.
The bacterium that causes stomach ulcers.
Mm-hmm.
I remember learning about that in biology class.
Yeah, it's a fascinating story.
But I'm curious to hear the story behind its discovery.
It was a rather controversial finding at the time.
It seems like it was a rather controversial finding at the time.
It was, yeah.
Yeah.
For decades, the medical community was convinced that stomach ulcers were caused by stress.
Stress.
The spicy food, excess acid production.
Right, the old conventional wisdom.
The old thinking.
But Barry Marshall and his colleague, Robin Warren, had a different idea.
They did.
They suspected a bacterium was the culprit.
Mm-hmm.
And they were right. I vaguely remember reading that Marshall even infected himself with H. pylori to prove his point.
He did. It's a famous story in the history of medicine.
Talk about dedication to science.
Yeah.
But before we get into that, you mentioned we have a Nobel lecture on this discovery.
We do. It's from Barry Marshall himself, who shared the Nobel Prize in Physiology or Medicine in 2005 with Robin Warren. For their discovery of Helicobacter pylori and its role in gastritis and peptic ulcer disease.
So this lecture takes us back to a time when the medical community was pretty skeptical.
It does.
Of the idea that bacteria could be the root cause of ulcers.
It was a paradigm shift.
What kind of challenges did Marshall and Warren face?
Oh, it was an uphill battle.
In trying to convince the world of their discovery.
Their initial findings were met with resistance and skepticism. Many in the medical community who were wedded to the old dogma about stress and lifestyle factors being the main culprits behind ulcers.
So how do they ultimately convince the skeptics?
Well, they presented compelling evidence from their research showing a clear link between H. pylori infection and the development of ulcers.
But perhaps the most convincing piece of evidence came from Marshall himself.
Yeah, that's right. He deliberately infected himself with H. pylori, developed gastritis, and then successfully treated it with antibiotics. Talk about a self-experiment.
Talk about putting your money where your mouth is. It was a bold move that ultimately helped change the course of medical history.
It was a game changer for our understanding of ulcers.
What were some of the key insights into H. pylori and its role in ulcers that Marshall's research revealed?
Well, one of the key findings was that H. pylori is incredibly well adapted to survive in the harsh acidic environment of the stomach.
It's got to be a tough place to live.
It is, yeah. It produces an enzyme called urease, which neutralizes stomach acid, creating a more hospitable niche for itself.
So it's like this bacterium brings its own antacid to the party.
That's a good way to put it.
What a clever survival strategy.
And Marshall's research showed that this H. pylori infection triggers an inflammatory response in the stomach lining, leading to gastritis.
Gastritis?
Which can develop into ulcers in some individuals.
This deep dive is highlighting the importance of challenging dogma in science.
It is, yeah.
It's a reminder that our understanding of the world is constantly evolving.
Absolutely.
And that sometimes the most groundbreaking discoveries come from questioning the status quo.
That's what drives progress.
Couldn't agree more.
Yeah.
Marshall and Warren's willingness to challenge conventional wisdom ultimately led to a paradigm shift in our understanding of ulcers.
It revolutionized the treatment of ulcers.
Transforming their treatment and improving the lives of countless people.
Countless people, yeah. It's a great example of how scientific discoveries can have a real impact on human health.
It's an inspiring story.
It is.
Of scientific tenacity. And the power of questioning assumptions.
A great reminder to never take things for granted. Always be questioning.
Indeed. Now, speaking of paradigm shifts. How about we explore the work of Elias James Corey?
Elias James Corey.
Who developed a revolutionary approach to organic synthesis called retrosynthetic analysis.
Retrosynthetic analysis. Okay.
Retrosynthetic analysis.
That sounds like a strategic and systematic approach to planning chemical reactions.
Yeah.
I remember organic synthesis being presented as a bit of a puzzle.
Yeah, it was a puzzle.
With chemists trying to figure out
right.
how to assemble complex molecules from simply starting materials.
Like a molecular jigsaw puzzle.
It is, yeah. And before Corey's work, organic synthesis often involved a lot of trial and error.
Right.
With chemists trying different reactions and hoping for the best.
Hoping for the best, yeah.
But Corey's retrosynthetic analysis brought order to the chaos.
And we have a Nobel lecture on this approach.
We do. It's from Elias James Corey himself.
Okay.
Who won the Nobel Prize in Chemistry in 1990.
Right.
For his development of the theory and methodology of organic synthesis.
Right.
Particularly his work on retrosynthetic analysis.
So this lecture takes us into the world of molecular design, where chemists use their knowledge of chemical reactions to strategize and execute the synthesis of complex molecules.
Like a molecular architect.
It is like an architect.
Yeah, planning out how to build these complex structures.
So Corey's lecture lays out the principles
of retrosynthetic analysis.
Okay?
Working back.
Which involves working backward from the target molecule.
So instead of starting from the beginning,
you start from the end?
They start from the end.
And work your way back.
Yeah.
You break it down into simpler and simpler precursors.
Yeah.
Until you arrive at readily available starting materials.
So it's like reverse engineering a molecule.
That's a great analogy.
You start with the end product and then deconstruct it to figure out the best way to build it from scratch.
Exactly. It's a very clever approach.
That's really cool.
And Corey's lecture highlights the power of this approach in simplifying complex synthetic problems.
Enabling chemists to create an astonishing array of molecules. From natural products to new drugs.
What are some specific examples of how retrosynthetic analysis has been used to create molecules that have had a real impact?
Well, one of the most notable examples is Corey's own synthesis of prostaglandins.
Prostaglandins.
These are hormone-like molecules involved in a wide range of physiological processes. From inflammation to blood clotting. Retrosynthetic analysis was key to planning and executing the synthesis of these incredibly complex molecules.
So this isn't just some theoretical exercise.
No, not at all.
It's a powerful tool that has led to the creation of molecules that have improved human health and well-being.
Absolutely. It's a great example of how chemistry can be used to solve real-world problems.
This deep dive is showcasing the amazing ingenuity of scientists in tackling complex challenges.
It is,
Whether it's developing powerful techniques like NMR spectroscopy or creating systematic approaches.
Like retrosynthetic
analysis.
It's all driven by a desire to understand and manipulate the world around us.
I couldn't agree more. And speaking of ingenuity, how about we delve into the work of Paul Lauterbur and Sir Peter Mansfield, who revolutionized medical imaging with the development of magnetic resonance imaging.
MRI.
Or MRI.
MRI.
Now, that's a technology I'm familiar with.
It's a very powerful technology.
It's incredible how we can create detailed images of the inside of the body without using harmful radiation.
It's amazing, isn't it?
It truly is. And Lauterbur and Mansfield's work on MRI was groundbreaking.
It was a major breakthrough in medical imaging.
They combined principles from physics, chemistry, and engineering.
A truly interdisciplinary effort
to create a technique that has revolutionized medical diagnosis and research.
It's had a huge impact on medicine.
And we have a Nobel lecture on this revolutionary technology.
We do. It's from Paul Lauterbur. Who shared the Nobel Prize in Physiology or Medicine in 2003 with Sir Peter Mansfield for their discoveries concerning magnetic resonance imaging.
So this lecture takes us back to a time.
It does.
When medical imaging was undergoing a major transformation.
It was a time of great innovation in the field.
With new techniques like CT scans.
CT scans, ultrasound.
And ultrasound also emerging.
Lots of new ways to see inside the body.
What were some of the key insights that led to the development of MRI?
I know it involves magnetism,
but I'm a bit fuzzy on the details.
Well, one of the key insights was Lauterber's realization that by varying the magnetic field across a sample, it was possible to encode spatial information into the NMR signals.
So by creating a sort of magnetic map,
essentially, yes,
different regions of the body would emit distinct signals that could be used to reconstruct an image.
Exactly.
It's incredible how they were able to bridge the gap between physics and medicine.
Yeah.
Using principles of magnetism and nuclear resonance to visualize the human body in such detail.
It was a truly remarkable achievement.
It's a testament to the power of interdisciplinary research.
I agree.
And the amazing things that can happen when scientists from different fields come together to tackle a challenge.
Breaking down those barriers between disciplines.
This deep dive is highlighting that interdisciplinary approach time and time again.
Yeah, it's a recurring theme.
It's a reminder that some of the most groundbreaking discoveries come from bridging the gaps between traditional disciplines.
It's at those intersections that the real magic happens.
Yeah. Okay, where to next on our scientific exploration?
How about we return to the world of economics?
Back to economics, and
explore Lawrence Klein's Nobel lecture on the development of econometric models.
Econometric models.
For analyzing the global economy.
Econometric models.
They're mathematical representations of economic systems. Using statistical methods to analyze data and predict future trends.
So it's like trying to capture the complex workings of the economy.
Yeah, it's a very ambitious undertaking in a mathematical equation. That seems incredibly ambitious. It is very ambitious.
And Klein's work was pioneering because he developed large-scale econometric models that could be used to analyze the entire global economy.
Not just individual countries or sectors. He was trying to model the entire system.
And we have a Nobel lecture from him on this topic.
We do. It's from Lawrence Klein himself, who received the Nobel Prize in Economics in 1980 for his work on creating these econometric models and applying them to analyze things like economic fluctuations and the impact of different economic policies.
So this lecture takes us back to a time when economists were trying to grapple with the growing complexity.
Yeah, the global economy was becoming increasingly interconnected.
Of the interconnected global economy.
And Klein's work was at the forefront of trying to understand how to model and analyze that complexity.
What were some of the key aspects of Klein's econometric models that he emphasized in his lecture?
Well, one of the key features was their comprehensiveness.
Comprehensiveness.
His models incorporated data on a wide range of economic variables, from consumption and investment to government spending and international trade.
So his models aim to capture the interconnectedness of the global economy.
Exactly.
Considering all the different factors.
He recognized that trying to isolate individual parts of the economy wouldn't give you an accurate picture of how it functions as a whole.
That makes sense.
And another key feature was their dynamic nature.
Dynamic nature.
Yeah, Klein's models didn't just provide a static snapshot of the economy. They aimed to simulate how it might evolve over time.
So they weren't just a picture in time.
No, they were trying to...
A model that could...
predict how the economy might change in response to different factors.
This deep dive is highlighting the power of mathematical modeling in science.
It is, yeah.
Whether we're talking about the nuclear shell model or econometric models, they give us frameworks for understanding complex systems.
Models are essential tools for scientists in many different fields.
Even if they can't perfectly replicate every aspect of reality.
Of course, models are simplifications, but they can be very powerful simplifications.
They're tools.
They help us to understand the essential features of complex systems.
Absolutely.
Yeah.
Okay, I'm excited to see where this deep dive takes us next.
How about we return to the realm of chemistry?
Back to chemistry.
Yeah. Okay. And explore Irving Langmuir's Nobel lecture on surface chemistry.
Surface chemistry. Surface chemistry.
That sounds like the branch of chemistry.
That's what it sounds like.
That deals with the interactions between molecules at surfaces. You got it. And it's a crucial area of study because those surface interactions are essential for a vast range of phenomena, from how catalysts work to the corrosion of metals to the behavior of biological membranes.
It's a lot of stuff.
Langmuir's work laid the foundation for our understanding of this important field.
And we have a Nobel lecture from him on this very topic.
Oh, we do. It's from Irving Langmuir himself.
Irving Langmuir.
Who won the Nobel Prize in Chemistry in 1932 for his discoveries and investigations in surface chemistry.
So this lecture takes us back to a time when chemists were just beginning to appreciate the unique properties and behavior of molecules at surfaces.
It was a new frontier in chemistry.
What were some of the key principles of surface chemistry that Langmuir highlighted in his lecture?
Well, one of the key principles was his model of monolayer adsorption.
Monolayer adsorption.
Which proposed that molecules adsorb to surfaces in a single layer.
Right.
Each molecule occupying a specific site on the surface.
So it's like a molecular puzzle?
Yeah, that's a good analogy.
With each molecule snapping into a specific spot on the surface.
It's a very ordered arrangement.
And Langmuir's work also revealed the importance of surface tension.
Surface tension.
The force that causes liquids to minimize their surface area.
Right. That's why water droplets form beads.
Yeah.
It's trying to minimize its surface area.
In governing the behavior of molecules at interfaces.
It's a very important concept in surface chemistry.
This deep dive is highlighting the importance of understanding how things work. At the most fundamental level.
Yeah, that's what science is all about.
Langmuir's work on surface chemistry showed that the behavior of molecules at surfaces can be vastly different from their behavior in the bulk of a liquid.
They behave differently at the interface than they do in the bulk.
This has huge implications for understanding.
It does.
Chemical and biological processes.
It affects everything from how reactions occur to how cells interact with each other.
And his insights have had a profound impact on various fields, from material science
to nanotechnology to biomedicine.
Okay, I'm ready for another scientific adventure. What's up next?
How about we delve into the world of biochemistry and explore Carl and Gertie Corey's Nobel lecture.
Carl and Gertie Corey.
On discovery and characterization of the enzyme polysaccharide phosphorylase.
Polysaccharide phosphorylase.
Okay. Are you ready for another mouthful of a name?
Yeah, I think I can handle it.
All right.
Polysaccharide phosphorylase.
That sounds like it's involved in breaking down carbohydrates?
It does, doesn't it?
You're on the right track. Polysaccharide phosphorylase.
Okay.
Or phosphorylase for short.
Phosphorylase.
Is a key enzyme involved in the breakdown and synthesis of glycogen.
Glycogen.
A complex carbohydrate that serves as a storage form of glucose in animals.
Okay, so we're talking about the enzyme that helps our bodies store and release energy.
Yeah, it's all about energy balance.
From carbohydrates, ensuring a steady supply of fuel for our cells.
It's a key player in metabolism.
And we have a Nobel lecture on this enzyme.
We do. It's from Carl and Gertie Corey.
Carl and Gertie Corey.
Who shared the Nobel Prize in Physiology or Medicine in 1947 with Bernardo Houssay for their discoveries relating to the catalytic conversion of glycogen, specifically their work on phosphorylase.
I remember reading about Gertie Corey. She was the first American woman to win a Nobel Prize in science.
That's right. She and her husband, Carl, were a remarkable scientific duo.
Yeah, I've heard of them.
Making groundbreaking contributions to our understanding of biochemistry and metabolism.
What were some of their key findings about phosphorylase? And its role in glycogen metabolism?
Well, one of their key findings was that phosphorylase catalyzes the breakdown of glycogen into glucose 1-phosphate.
Glucose 1-phosphate.
Yeah, a crucial intermediate
in glucose metabolism.
And this process is reversible, right?
It is, yes.
All right.
So phosphorylase can also help build glycogen back up from glucose 1-phosphate.
So phosphorylase acts like a molecular switch.
That's a good analogy.
Controlling the flow of glucose into and out of storage.
It's regulating the balance between storage and release.
Depending on the body's needs.
And the chorus also discovered that phosphorylase exists in two forms.
Two forms?
An active form and an inactive form.
So there's like an on switch and an off switch.
They found that hormones like adrenaline and insulin play a crucial role in regulating which form is dominant.
So the hormones are controlling the switch.
Essentially, yes.
Yeah.
Effectively controlling the storage and release of glucose.
This deep dive is showcasing the elegance
and
complexity of metabolic pathways.
It's a very intricate system.
Yeah.
But it works beautifully.
It's fascinating how our bodies have evolved for such sophisticated mechanisms.
To manage our energy balance.
Ensuring a steady supply of fuel
for all our cells.
And the chorus's work paved the way for a deeper understanding of metabolic disorders.
It did, yeah.
Like diabetes.
Where that delicate balance is disrupted.
Right, right.
It's a great example of how basic research can lead to important medical advances.
It's remarkable how all these discoveries build upon each other.
That's the beauty of science.
Revealing not just the individual pieces, but the intricate web of interactions that keep our bodies functioning.
It's all interconnected.
It is. And speaking of interconnectedness, our next stop takes us back to the early days of a technology that has truly transformed how we communicate and connect with the world. Wireless telegraphy.
Wireless telegraphy.
Wireless telegraphy. That sounds like the precursor to modern radio.
It is, yeah.
I can't imagine a world without instant communication. It's hard to imagine, isn't it?
It is.
But back in the early 1900s, the idea of transmitting signals through the air without wires was revolutionary.
It was a huge leap forward.
It was.
Yeah.
And we have a Nobel lecture from one of the pioneers of this technology. Carl Ferdinand Braun.
Carl Ferdinand Braun.
Wow. So we're going back to the very birth of radio communication.
That's amazing. What were some of Braun's key contributions to this field?
Well, one of his key contributions was developing a much more efficient and reliable transmitter for wireless telegraphy.
So he was basically improving the way those early radio signals were generated and transmitted.
He figured out how to use a coupled circuit design to generate those high-frequency electrical oscillations that carry the signal.
What about receiving those signals?
I
imagine that was a challenge as well.
You're right. And Braun also played a key role there. He developed something called a crystal detector.
A crystal detector?
Yeah. Which use the unique properties of certain minerals, like galena, to convert those high-frequency radio waves into audible signals.
So his work helped to make those early radio receivers more sensitive.
It did,
and capable of picking up those faint signals from afar.
Exactly. And his work on crystal detectors laid the foundation for even more sophisticated radio receivers, eventually making it possible to transmit and receive not just simple dots and dashes, but also voice and music.
It's amazing to think how far we've come.
It is.
Since those early days of crackling radio signals. This deep dive has really brought to life the stories of scientific ingenuity and perseverance throughout history.
It's been a fascinating journey.
It's inspiring to see how those early discoveries have paved the way for the technology we take for granted today.
It really shows how science builds upon itself over time.
Well, we've reached the end of our deep dive.
We've come to the end.
What a journey it's been. We've explored a mind-boggling array of scientific discoveries. From the microscopic world of ion channels to the vast expanse of the cosmos.
It's been a whirlwind tour.
What stands out to you as the most significant takeaway from this exploration?
Well, for me, it's the realization that science is not a collection of isolated facts, but a dynamic and interconnected web of knowledge.
It's a constantly evolving story.
It is. Each discovery builds upon others. Sometimes in unexpected ways.
Absolutely.
Leading to new insights and innovations.
I completely agree. And it reinforces the idea that progress in science often comes from challenging assumptions.
Absolutely.
Asking those what-if questions. And being open to new perspectives.
Even when they seem radical or unconventional.
Right, like those scientists we were talking about.
We saw that with Barry Marshall, who challenged the long-held belief about the causes of stomach ulcers. And with Sidney Altman, who overturned the dogma that only proteins could act as enzymes.
Those stories are a powerful reminder that the pursuit of knowledge requires not just brilliance, but also courage and a willingness to go against the grain.
To think differently.
Absolutely.
To challenge the status quo.
Now, before we wrap up completely, you mentioned there were a couple of intriguing details from the sources.
A couple of little tidbits.
That we didn't have time to fully unpack.
We didn't.